here I am. Hi, everybody. My name is Alejandro. Oh, we got a couple of more people coming in. So we got Westboro, Massachusetts. We got Janice from New Jersey. Welcome, welcome. All right, so as I mentioned before, real quickly, my name is Alejandro. I'm one of the STEM educators at Liberty Science Center, part of a big team of the STEM department. And so you may have recognized me from previous sessions of the Live from Surgery, whether it's the cow eye dissection, the sheep brain dissection. I had a couple of our surgeons from the Live From program chime in and show a little bit of their surgeries. If this is your first time here, welcome, welcome. All right, so don't worry about the ingredients just now. I'll post them up later on, so we'll have plenty of time to do so. But I would like to welcome you all. This is actually our final Live From Surgery live stream session with you all. So you may also have seen Miss Caroline doing some of the streams with some other topics. She did cardiac risk factors. She talked about bones. And so Miss Caroline is also in the comments moderating, and alongside with Miss Rosa. They'll be happy to answer any questions that you may have during our session. So definitely do say hello to them in the comment stream, and they'll see them as well. And they'll probably actually see it way better than I will, because I got a lot of things going on my side. So if I don't get to your questions, Miss Caroline, Miss Rosa will be more than happy to help you out and answer those questions for you. All right. So before we begin, I also want to give a special shout out to all the members and all the contributors that are helping keeping all this Live From sessions running. So not only you might be familiar with the planetarium, with the animal updates. I know the animal updates ended last week. We had our LSC Junior, which ended yesterday, and then we still have some trivia going on. So without their contribution, we wouldn't be able to give you all of this content from Monday through Friday. So please feel free to click on that donate button that should be highlighted at the bottom of the comments thread, and we would greatly appreciate it. All right, so let's begin with our lesson, because obviously the most important part of this program is to do this experiment, correct? All right, so we are going to be doing some DNA extraction in our homes. All right, so before I begin, I have to really canvass and review how much you know about this topic. So drop in some comments, some facts about what you know of DNA, and I'll give you some more information on that. But in the meantime, I'll go through the stream, give you some special shout outs, but let me know what you know about DNA. All right, so we got Connecticut, we got Scotch Plains going on here. Welcome, welcome. We got Aditi from West Windsor. All right, all right. Saria is going to 5th grade middle school. Beautiful. Congratulations. I know someone mentioned deoxyribonucleic acid before, which is beautiful. So what do we know about DNA? So Connor says everything that is alive has DNA. Beautiful. We also have DNA is mostly known in a double helix form. Decides how we look. Everything has DNA. DNA has both your parent cells in it. Excellent, excellent. It is located in our cells. Beautiful. And I see a lot of you are saying you're going up to a new grade. I saw a couple of you going to 5th grade, 4th grade, 1st grade. Congratulations. We got Columbia, South Carolina, moving on up, being 48 years of the School of Life. Congratulations. Congratulations on moving up in the School of Life. That's a great one. And it's the makeup of life. So we got New York City in there as well. Welcome, welcome. All right, so let's talk a little bit more about DNA. So a lot of you are pretty much on point. All right, so we got DNA, which stands for deoxyribonucleic acid. All right, and as a lot of you mentioned, it is found in every living thing. And since a lot of living things are made up of cells, DNA is found within the cells. So DNA, the material itself, is pretty much a genetic recipe. It's all this information tightly packed into this small bit of material, and it pretty much dictates how an organism lives and a lot of the different functions that happen within the body. All right. So a lot of it usually determines on how we look. I have brown eyes. I used to have curly hair. Now I'm just all bald. All right. Also, you know, with taste. Okay. So that's a couple of other things in terms of what DNA exhibits. All right. So real quickly, DNA, actually, when you look at it down to the nitty gritty, it is made up of 4 code blocks. We call them bases. All right. So we have adenine, thymine, guanine, and cytosine. So ACTG. So dependent on the order of those different code blocks is located on that certain section of DNA, that really determines the information that we need. So a lot of the times, not only does it determine our eye color, but also helps with the processes that happen within our body. It makes all these different types of proteins to help our body function. So those proteins are moved over to tissue, tissue to organ, organ to organ systems, all right? So it helps with all of those different functions. So DNA, as a lot of you mentioned, it's located in the cell, particularly in the area known as the nucleus. We kind of call that as the nerve center of the cell. All of that important information for that cell is located in the nucleus. When we peel back that nucleus and kind of magnify further in, we have these little things floating about called chromosomes. All right, so chromosomes are the larger aspect of DNA, where DNA is kind of pretty much tightly packed and coiled around. All right, and so when we go further in, that's where we get that double helix shape. And then we can actually, like a zipper, unzip that, get that piece of code, and make whatever we need in order to function. All right, beautiful. Okay, so I know a lot of you had a lot of awesome thoughts about DNA, so that's great. So what we're going to do next is we're going to jump into the experiment. There is not much going on in terms of teaching you all, because you all seem to be experts in the topic of DNA. So why don't we just jump into the experiment itself? All right. So let's extract some DNA. All right. So I know a lot of you wanted to look at the materials list, so I'll actually post them up right here. Okay. And I'll give you about a minute to gather all the materials that you may be missing. All right. So I'll go down the list. So you need about a ½ teaspoon of salt. You need a 1/3 cup of rubbing alcohol. You need a 1/3 cup of water. 1 tablespoon of liquid dish soap. So that's pretty much the soap that you use to wash your dishes at the kitchen sink. Okay. Metal strainer. So I actually have one here. But if you don't have a metal strainer, what would work best is if you have a coffee filter or a cheesecloth or paper towel. And what you can do is you'll use that as your barrier, as your filter. All right, and you'll be using that later, but if you can get yourself a big cup, and that way you can actually, if you have a partner, which is perfect, all right, you can actually get one partner to hold on to the paper towel or cheesecloth or coffee filter, and the other partner is going to hold on to the cup, and that's where we do our filtration. A couple of glasses, one for getting that filtration, another one for mixing. 3 strawberries. I'll explain a little bit more as to why strawberries are a little bit more important. Okay. Ziploc bags and toothpicks. All right. So let me show you some of what I have here. Okay. So what I have here is some strawberries. I kind of already made some of that. A little bit of a cooking show action going on here. But I'll walk along with you if you have strawberries through this part right here. I have my water right there. So I'm actually using distilled water, but it's okay. You can use tap water. Really doesn't matter. Actually, I just have a bunch of distilled water lying around. I have my salt in my little beaker there. I actually have my dish soap right here. So if you're wondering, this is actually like a medicine cup from like cough syrup. So if you actually fill it up to a good portion of that, so I don't know if I can show you here. So let me show you right there. So right here is the line where it's like 20 milliliters. So I kind of filled it up just underneath. And so a medicine cup full of soap would actually do for this experiment. I got my Ziploc bag, and I got my strainer as I showed you. Oh, and my isopropyl alcohol. So I actually have it on the side here, right there. So I got my alcohol there, my rubbing alcohol. All right, so I'll give you about 1 more minute. So I'll post up the directions up there, the materials list again. All right, and if you are ready, definitely let me know down in the comments if you are ready. If a majority of you are ready with all your materials, then we'll begin. But I'll come through the comments again, see what we got going on here. And I see that Ms. Caroline, Ms. Rosa were kind enough to lay down the ingredients list on the comment stream as well, so definitely check it out. All right, let's check out. All right, so Alex from New Jersey. Oh, LSC member. All right. Going to the 2nd grade. Congratulations. Sophia's going to the 3rd grade. Congratulations. We also got Kate going to the 3rd grade as well. 6th grade, 1st grade. Oh, Brooklyn, New York going to the 5th grade. Excellent, excellent. All right, so I see a lot of you are already coming in saying that you are ready. So let's begin. All right, so the first thing that we have to do is put the rubbing alcohol in the freezer. We need that rubbing alcohol to be as cold as possible. All right, so right now, if I were to show you my little setup here, I actually had my rubbing alcohol in the freezer for a bit. And now I have it in this little vial, and I have kind of like a lot of ice wrapped around it to keep it as chill as possible. So that's why it's on the side there. Okay? But put that alcohol in the freezer. All right, so, oh, Miriam's going to the 5th grade. Drake's going into 4th. Beautiful, beautiful. Congratulations. All right, so now that you have the alcohol, the rubbing alcohol in the freezer, I'm assuming you're probably running back to the computer. You're probably having your lab partner, probably an adult, running back. Claire's going to the 5th. Beautiful, congratulations. All right, so here's the next step. We're gonna mix the salt, the water, and the soap in a 2nd cup. So I actually have mine in the measuring cup, so I'm actually gonna do all the mixing there. And we're gonna consider this our extraction liquid. All right, so that's gonna be our extraction liquid. So I'm gonna pour all of my ingredients in there. So I got my dish soap in there, so I'm gonna refocus. There you go, so everyone can see nicely. And we got salt. All right, so I'm gonna pour that in there, and now I'm gonna mix. I'm gonna start stirring. All right, so this is what we're gonna consider from this point on. We're gonna consider it our extraction liquid. So once again, you're going to use that cup for mixing that you designated as a mixer. You're going to mix your water, your salt, and your dish soap all together. And pretty much stir it until a good portion of your salt is dissolved. Do I know if we share DNA with bananas? Yes, that's absolutely true. So we actually share DNA with a lot of organisms. So I'll talk a little bit more about that in a little bit. So now that we have our extraction liquid, all right, we're going to move on to our next step. So step number 3 is cut off the tops of the strawberries and cut them into halves. Now, it's not recommended for you to cut them in halves. It's going to be easier for the next step that you're going to be doing. Oh, so what did you miss after the bananas? Oh, not the bananas, the freezing of the alcohol. So you're gonna mix your soap, your water, and your salt in 1 cup that you're gonna designate as your mixing cup, and you're just gonna stir it all together and mix it all together, and that's gonna be your extraction liquid. So once you're done with that, we're gonna set that aside and we're gonna move on to our strawberries. All right, so you notice I already have some of them cut in half. It's not necessary for you to cut them in half. It'll be easier for the next step that we're going to be doing. But I still have one that I did not do so much. For the water, you're going to do 1/3 of a cup of water. For the salt, I believe it's a 1/2 teaspoon of salt. So I actually have the measuring tools here. Oh, actually, it's right over here. So the salt will be 1/2 teaspoon. All right, so 1/2 teaspoon of salt. You're gonna have 1 tablespoon of soap, just like so. You're gonna mix it all together into that cup. All right, so I'm gonna pull off the top of my strawberry here. All right, so let me maximize the video there. Do you need to peel the kiwi? Yes, if you need time to peel some of that, you should be fine. We're looking for the middle part, but it can be interesting. You don't have to cut the skin. It'll just be a little bit longer for your extraction. But yes, you should recommended peel that kiwi back. If you have oranges, peel that orange, all of that skin, because what you want is the inside. So with strawberries, it's easier because you don't have to peel anything. So I'm going to cut this in half. It's not recommended once again. So be very careful if you want to cut your strawberry. Always ask an adult to cut it. If you don't have an adult to do so, then you're going to leave it whole. It should be fine. All right, so here's our next step. So now that we have our strawberries all set, we're going to put the strawberries in the plastic bag. And this is the fun part. This is actually my favorite part of the whole thing. We're going to mash the strawberries for 2 minutes. Okay? So, I'm going to put all my strawberries into my Ziploc bag. Hopefully I don't drop them to the floor. All right, excellent. So here we have our bag of strawberries right here. Okay? So, this is crucial. This is important. When we're going to mash our strawberries, we want to make sure that when we seal our bag, there's not any air bubbles in there. We want to make sure that there's no air. You're going to escape all the air in that bag as best as you can. All right, even if you got to put it on the table here. So let me get rid of my dish that houses the strawberries. All right, you want to make sure that all of that air is completely gone. All right, because when we're mashing our strawberries, we don't want our bags to pop and cause a mess. All right, so here we go. So I'm going to start mushing it all up. What you want to do is try and grab all that pulp and all that juice all the way to the bottom as best as you can. So pretty much 2 minutes is safe because that kind of mushes all of that fruit down as best as you can. You want to make sure that it's pulverized and mashed as much as you can. There's not many large chunks left. All right. Sure. No, you did not miss that step. So, all right, so I'll recount all the steps for you all. All right, so as I'm mixing, I'll go back to the beginning. All right, so step 1 is put the rubbing alcohol in the freezer. All right, so go ahead if you already did that step, great. Have your lab partner, or you can actually run to put that alcohol in the freezer. All right, so you're going to need 1/3 of a cup of alcohol, rubbing alcohol. Okay? Next step. We got the salt, water, and dish soap. You're going to mix it all in a cup. You're going to stir it. You're going to stir all those ingredients together. All right, just like how I did it here. So I'm not using my measuring cup anymore. So I mixed it all in there. All right, this looks beautiful. So you can see it kind of, all that goop in there, right in there. All right, so once you've done all that stirring, you're gonna get whatever fruit you're working on. Strawberries, just peel the green tops off. You don't have to cut it in half. You can actually do it whole and do the mashing like so. If you have kiwis or oranges, you might want to peel all that skin, outer skin off. If you're using apples, you should be okay with the skin attached. Bananas are pretty easy to mash. So just pretty much cut small pieces of that banana and stick it right in there. All right. So that's the third step. Now, this is the next step. Hopefully everyone is up to speed. Next step is, oh, we already did that part right there. So the next step is we're going to add the newly created extraction liquid. So that's the liquid right here. We're going to place it in the bag with the mashed strawberries. All right, so be very careful. It's going to get messy. So remember, you want to make sure that you have no air in your bag. Because when you start mashing and mixing all that together, you don't want your bag to pop and have a mess. All right, you can definitely try grapes. This is all the beauty of the experiment. So if you are using different fruits like grapes, kiwis, oranges, bananas, let us know. Find us on social media. We have Instagram. You can leave it on Facebook as a comment, Twitter. Take photos. We want to see what your results are. Beautiful question about why strawberry. So strawberry is considered octoploids. So that means that they have a lot of DNA found in one strawberry. Humans are diploid. So that means that we have one set of copies of DNA material from our mother. We have one set of copy of DNA from our father. So we have 2 sets of DNA from our parents. So that makes us diploid. Diploid means 2 of the material. Octoploid means 8 altogether. So that means strawberries have 4 copies from, the mother, 4 copies from the father. All right, in relative terms, when we're talking about fruits and flowering plants. All right. Okay, so here we go. We're gonna pour that into our bag. So hopefully everyone is up to speed. So be very careful. So I'm gonna pour all that good stuff in there. All right, so there we go. Look at that beautiful mixture. All right, so once again, make sure that all that air is not in the bag. You don't want your bag to pop. All right, so now you have that stuff there. So all you have to do is the same thing. Just kind of mash it around. Mix that all together. All right. So, our concoction here, our extraction liquid, the reason why we have all those different ingredients in there is that they all play a role in our DNA extraction. So soap, I'm sure you get a lot from your parents or from your adults, that you always need to wash your hands. Soap is great at destroying cells. So whatever germ, whatever microbe, whatever pathogen that might be on your hands, might be on surfaces, you use soap to pretty much eat away at the outer membrane, the cell membrane. So that way, all of that soap can actually penetrate inside the cell and destroy it. Okay? So that's what the soap is doing right now. So all the strawberry cells are being broken down by the soap. So all of that cell membrane, those strawberry cell membranes, are being broken down by the soap. Now the salt is responsible for breaking down the proteins that is keeping the DNA together. So as you remember, everyone's mentioning about the double helix and all that, and that formation, that beautiful twisted staircase look to it. So the salt is actually attacking all those different points of the DNA that kind of keeps it all twisted and wound up. It's loosening it all up, so that way we can extract that DNA much better. Okay? So I think about 1 minute has passed. All right? So there it is. There is my concoction. All right? So the next step is we're going to pour all this into a cup, but I have a strainer. So we're going to filter out all of that big pulp of strawberry. What we just want is the liquid portion of our mixture. Okay? So, I have my cup here. So let me go right there. So I have my cup. Hopefully it doesn't overflow in this cup, because I measured it before. It should be large enough for our concoction. I'm going to place my strainer here. So at this point, what you should see, and I actually have some paper towels just in case I made a mess. So the whole idea of the paper towel is to go like this above, and then you're going to pour that mixture in. And once you have all that mixture in, you're going to pretty much try and close it up and squeeze as much of that liquid into the cup. All right. So, let's do the straining. So here we go. So I don't have a lab partner, so I'm going to do my best to do this on my own. So there we go. Wish me luck. All right, so here we go. I'm going to pour it right in. All right. Look at all that. All right, so I kind of made a mess, which is okay. That's all right in science. Science, you know, you take chances, make mistakes. That's how you learn, and science can get very messy, which is okay. All right, so luckily this mat for my document camera is disposable, so I don't have to worry so much. So it looks like I have a good portion of this filtered out. So I'm going to bring back my measuring cup. I'm going to do a quick switcheroo. There we go. So there is my extracted strawberry liquid. All right, so that looks great. All right, so I'm just going to fix up here. I got some strawberry gunk here. All right, so I'm just going to do this. All right, beautiful. We're almost done. We are almost there. Okay? So, this is your moment. If you haven't done so, well, I should say, if you probably already placed your alcohol in the freezer, this is your time to go grab that alcohol. It should be cold enough for this next step. Okay, so what we're going to do next is we're going to slowly pour our rubbing alcohol into the glass. So, here's a little bit of science lingo, all right? What we call that process is decanting, all right? So decanting is pretty much putting both glasses together and slowly pouring it in. All right, so I'm going to do my best to do it under the document camera. If I can't do it, then I'll do my best to do it right here in front of the webcam, okay? So this is a very important step because you don't want to mix the alcohol with the liquid that you already collected. what you're going to see is you're going to make two layers. You're going to make a layer of that strawberry liquid and then a clear layer of rubbing alcohol. All right, so here we go. So I'm going to take out my chilled rubbing alcohol. All right, and we're going to decant our alcohol into our mixture. So you notice that I'm kind of, actually let me see if I can do it this way. So you notice I'm kind of slowly tipping forward together. All right, it could get a little messy. So once you start pouring, you got to commit. You got to let it continuously pour. All right, so here we go. Very slowly. So I am making a little bit of a mess, but that's okay. It's only alcohol. I do have paper towels here at the floor because I know I was going to make a mess. So as you can tell, I'm the one team member in STEM at LSC that tends to get very messy. It's not my colleagues, Ms. Rosa, Ms. Caroline can definitely tell you, they definitely do not like taking up the lab after me because I make a big mess. All right, so you should see something happening quite immediately. You don't see it very well here on my webcam, so I'm going to place it back onto my document camera. All right. So you don't see it very well there. Let me see if I can. So with the lights off, you can sort of see it. You see like white stringy stuff popping up. Let me see if my little toolkit here can make it pop more. So you notice all that white stringy stuff already coming out in our mixture. So let me zoom in. Ah, there we go. So now that I zoomed in, beautiful. So I'm going to take off the light. So there you go. See all of that stringy stuff? You can actually see the DNA getting pulled out from that mixture. All that white stuff is strawberry DNA. So the way that this works is that strawberry is, it's not strawberry, I should say, for our DNA, the material itself, regardless of what organism it comes from, the way that it is formed, it is considered insoluble. So that means that it will not dissolve in a medium. All right, so what we're seeing here is that the alcohol cannot dissolve the DNA. All right, what DNA will do as a result is that once it gets pulled out from the liquid mixture, it's going to find itself. It's actually going to attract to each other. It's going to make these small little clumps. And that's what the stringy stuff that you see going on in our alcohol layer. All right. The reason why we use rubbing alcohol once it was frozen, because actually it never freezes, it actually becomes super, super cold, is that it helps with the extraction process much quicker. All right. Someone actually brought up an excellent question. Geetha says, is it the same way human DNA is extracted as well? Very excellent question. So, in a way, it is a similar process. Obviously, you're going to be using different chemicals, different reagents, different substances to help pull that DNA from whatever sample you collected it from, whether it's a hair follicle, skin cells, inside your cheek, maybe the back of your throat. All right. It's a similar process in terms of extracting DNA. So what I'll do next is I have a pair of tweezers and I'm going to pull it out and see if I can get some of that DNA out. There should be enough clumps for me to pull it out, but if not, you have all of that DNA there. Okay? So let me pull it out. of our mixture here. So let's see if I can get some. It's a little bit hard, but you can see right at the tip of my forceps here, my tweezers, there is some DNA at that very tip. So you can use 1 or 2 toothpicks to collect all of that if you can. It's going to be a little difficult, but I think because toothpick is wood, it might cling to the wood a little bit better than my tweezers, but there is that DNA. All right. So that's the entire experiment. So I know one of you asked, you know, is this the same process that we do with human DNA? Absolutely. It's a very similar process. Like I said before, it's just different chemicals, different materials to extract that DNA from the cells. Okay? So, a lot of you have already been mentioning that you are using different types of fruits. So, my challenge to you is you can repeat this experiment with other fruits. Try bananas. I know someone asked about kiwi. Someone asked about raisins. Raisins, you gotta hydrate them so they can become grapes again. So grapes are a little bit better because there's still some liquid in there, some juices. You can also try cherry tomatoes. I heard someone did cherry tomatoes that came out just fine. Another one is wheat germ flakes. So you can find those at the supermarket as well. All right. If you use any other type of fruit, I know someone mentioned oranges. All right. Please let us know. Definitely post those pictures. Show us those results. I would be more than happy to check them out. I would love to see those results. All right. As I mentioned before, strawberries are just easier to get DNA because they're octoploid. They have a whole bunch of DNA in that one strawberry. All right. They have 4 copies from the mother, 4 copies from the father. So that's 8 copies altogether. Humans, once again, are diploid. 1 copy from the mother, 1 copy from the father, I should say. Now, what are some of the applications for DNA? All right, we can do a lot of DNA testing. We can actually, a lot of you probably have relatives that are huge fans of genealogy tracing. So that's those that kind of do a cheek swab and send it in, and you can find out, you know, where your genealogy comes from. So you can do that. You can actually trace it based on DNA because certain segments of DNA remains consistent within a family tree. So it's very easy to trace it back as far as you can, depending on record keeping, so on and so forth. Archaeology is the same way. We can find archaeological sites. If there's any evidence of DNA that can be collected, they can actually build a profile of that civilization that lived in that area, or an individual that lived in that area. Genetic counseling. So it's actually pretty big in the few recent years where they can actually run a DNA fingerprint and get a nice little genome of a baby before they're born, so they can have a genetic profile to see the health of the child. So that way they can get ready if there is any things that they need to be aware of. Forensic science. DNA is used a whole ton in forensic science. Collecting evidence like hair follicles, there might be blood in the crime scene, they can collect that. They'll do a whole bunch of that at the scene of the crime. They collect all that evidence, run it through the lab. And viral testing. So viruses, it's up for debate whether they're considered living things or not. I personally don't think they are living things, but they still have genetic material. Some viruses have a single strand called RNA, which our DNA molecules can produce in order to make those proteins and that information, or some of them do have DNA. Very rarely will they have DNA. It's mostly RNA. We can actually run tests where we can get a nasal swab or a cheek swab or a throat culture and run certain tests to see if a certain segment of that RNA material belonging to that virus is found within a patient. All right, so I will do the last 3 minutes, see if you have any questions that you may have. I know we're almost out of time, but do check us out at lsc.org for LSC in the House content. I know a lot of you had trouble keeping up. There will be a video of this session so you can see for later use. All right, and definitely do donate. The donate button is on the bottom there of the comment stream if you are able to. We greatly appreciate it. Once again, a special shout out to our members and the contributors who have already supported our whole initiative here of live streaming. As I mentioned before, this is my final live stream. Last week was Ms. Caroline's final live stream. So it would figure it'd be appropriate to kind of bookend it since I started the very first live stream initiative with many members at the Science Center that is responsible for putting all this together. We had our very first live stream during our times here back in March. I'm bookending it now here, so don't think of it as a permanent end. It is just a season finale. Hopefully, we do reopen very soon. So hopefully I get to see you all. Come visit us. If you do see me walking about, and most likely I will, definitely don't be afraid to yell out my name and say hello within social distancing rules. All right, but are there any questions left that I may be able to answer that Miss Rose and Miss Caroline may not have answered? You're welcome. I see a lot of this was great. Beautiful, beautiful. Can you use a pear? Yes, you can use a pear. So you can get a good number of DNA from any type of fruit. Strawberries just happen to be better because of that makeup of strawberry. Actually, I think we got some more DNA floating up top, so I'll show you that. Should be easy to see now on my screen. Have I done any experiments with dino DNA? No. I wish I did. I unfortunately do not have the credentials to do so. But I do know a couple of individuals that have done it, which, you know, they say it's pretty cool. So there is pretty much all my DNA. So if you let it settle and let it do its work, you can see all of that DNA come up at the very top layer. What are some other fruits or stuff you can use? Excellent question. You tell me. So definitely try it out. Try out any fruit. So if it's like an orange or kiwi, normally fruits that you would have to peel, make sure you peel that and just get the interior of the fruit. And let us know. I forgot to mention, look for us up in our social media. Hashtag Everything Liberty Science Center. Also, if you want to say hello to Ms. Caroline, to myself, Mr. Kango, because I know he had his last live stream, or anyone else that's been doing the live streams, you can easily send us a message of some of the experiments that you've been doing to our social media. And my colleague Jeremy will be more than happy to forward any of those greetings and all those test results, those experiment results to us, and we'll definitely be happy to respond to them. Are all fruits and veggies octoploids? Very good question. As far as I know, strawberries is unique in that manner. For other fruits, I'm not exactly too sure. Is all DNA white? Very good question. Based just on visual senses, it is white, only because of the alcohol, I believe. But yeah, just by makeup alone, if you do the wheat germ, if you do the bananas, you'll get white DNA. There's substitute for rubbing alcohol. As far as I know, there isn't. Alcohol is one of the more crucial ingredients that you need to have to make this experiment consistent. Vegetables, I do not know. Vegetables, I'm not too sure if it's doable, but like I said, try it out and show us the results, and we can add that to the list. All right, so that's the last of our session here. So once again, this is Alejandro signing out. Hopefully I get to see you once we reopen and you come visit us. But I had so much fun connecting with you all week to week with Miss Caroline. And I hope you all enjoyed all the anatomy, all the live from related surgical content that we gave you. And enjoy the rest of your summer. Enjoy.